Okay, so now moving on to our second macromolecule, and that's going to be lipids. They also contain carbon, hydrogen, and oxygen, but not in any specific ratio like we saw in carbohydrates. They also have a little bit less oxygen than when we were talking about carbohydrates previously. If we're talking about more complex lipids, they also can incorporate phosphate into their structure. If you're thinking about lipids in general, go back to the phospholipids that make up the plasma membrane. So they're not soluble in water. We have a couple main types of lipids. We have triglycerides, which are the major form of stored energy. We have phospholipids, which again are those components of the cellular membranes. We have steroids, which an example of a steroid is cholesterol, which is also a component of the plasma membrane. And we have eicosanoids. And I'm only mentioning it briefly here. It's a very diverse group. It includes things like prostaglandins that help with blood clotting and regulating blood pressure. But just kind of understand that that's a type of lipid, and these are your so is steroids, phospholipids, and triglycerides. So here's a triglyceride structure shown to you over here. Basically what it is it's a glycerol. So that's this rectangle box right here. With 3 fatty acid chains. So this is a fatty acid chain, so is that one and that one. So that's why it's called a triglyceride. 3 fatty acids attached to a glycerol. When we're talking about triglycerides, typically we call them fats when they're solids, and we call them oils when they're liquids. And again, they're the major form of stored energy in the body. When we oxidize triglycerides, they can provide huge amounts of energy. So just like with any of our macromolecules, we have the same general reactions in order to build triglycerides up and to break them down. So every time we want to, again, link something to something else, we have to remove water. So every time we want to link one fatty acid chain to the glycerol unit, we have to remove water. So that is a dehydration reaction. And because we have 3 fatty acid chains in a triglyceride, that means we actually have 3 dehydration reactions in order to attach all 3 fatty acid chains to the glycerol unit. If we want to break it down, that would mean that we would need 3 hydrolysis reactions, 1 for each fatty acid chain, in order to remove it. So when we're talking about triglycerides or fatty acids, we typically group them in either being saturated fatty acids or unsaturated fatty acids. If you remember, one of the important things of organic molecules is that they all contain carbon. A unique feature of carbon is that it's able to form 4 covalent bonds. So here's a carbon molecule, and here's 4 covalent bonds. We say that the carbon is saturated if carbon forms 4 individual bonds. So saturated fatty acids means that every carbon in this fatty acid chain is making 4 different bonds. And I know you haven't had organic chemistry yet, but basically this is how carbons are drawn in organic chemistry. So one line, each one represents a carbon. This is another carbon right here. That's a carbon, that's a carbon, that's a carbon. What's not drawn when we're talking about, again, organic chemistry is hydrogens. You just assume they're there. So there's technically a hydrogen here, a hydrogen here, a hydrogen here, and a hydrogen here. No, you don't need to know that. I'm just showing you this so you can see that this carbon is making 4 different bonds. 1 to a carbon here, 1 to a carbon here, 1 to a hydrogen here, and 1 to a hydrogen here. So that's why we say these fatty acids or these triglycerides are saturated, is because every carbon in the fatty acid chain is bonded to 4 different things, meaning that we don't have any double bonds at all. Because our carbons are bonded to 4 different atoms, they make their fatty acid chains very linear. So if you see this, it's a nice straight structure, all of these. Because we have that nice straight structure, triglycerides can stack very, very neatly at room temperature, which means saturated fatty acids are solids at room temperature because they have saturated carbons and they don't have any kinks in their fatty acid chains, and they can stack nice and neat. In contrast, if we're talking about unsaturated fatty acids, that means that every carbon is not necessarily making 4 unique bonds. In some cases, we're going to have a double bond. And if we have a double bond in a fatty acid chain, it's going to cause that fatty acid chain to kink, and therefore we can't stack nice and neatly all of those triglyceride molecules. So that is why our unsaturated fatty acids are liquids at room temperature. We also have trans fats. And when I was showing you earlier, if we're talking about a double bond between a carbon, we can have hydrogens. If hydrogens are in opposite position to each other, we say it's trans. If they're on the same side, we would say it's cis. You don't normally get trans fats in naturally occurring products. That is a result of undergoing processing of things like margarine and things like that. It happens in the process when it's being manufactured itself. So moving on to phospholipids, which again is another type of triglyceride. Basically, they're modified triglycerides, so structurally very, very similar, except instead of having 3 fatty acid chains, they only have 2 fatty acid chains. And we have a phosphate group that actually replaces the 3rd fatty acid chain. So we already talked a little bit about this when we talked about the plasma membrane of a cell. So the head of the phospholipid is going to be polar or hydrophilic, meaning that it loves water, and tails are going to be nonpolar and hydrophobic. So again, here's some more. Polar head, 2 fatty acid tails, and a phosphate group. Another type of lipid are what are considered a category called steroids. One prominent example of a steroid is cholesterol. And this is the structure of cholesterol over here. And it is 4 very rigid ring structures linked together. We don't have any fatty acid chains. We don't have a glycerol. We don't even have a phosphate. So this is why when we did categorize our 4 macromolecules and stated the monomers for each macromolecule, that I said lipids do not have a true monomer. Because if we look at this example of a steroid, it's structurally completely different than what we saw with a triglyceride or a phospholipid. So no true monomers for lipids because it's such a broad class of different molecules.